Hi, so my name is Sian Cully and I'm going to be giving you two lectures on super resolution microscopy, very basics of the technique, so you kind of get to grips what this whole family of microscopy methods are all about. I personally am currently a postdoctoral research associate at the MRC Laboratory for Molecular Cell Biology at University College London, and I've been doing super resolution microscopy for getting on for the best part of a decade now, which is slightly worrying and makes me feel quite old. Okay, so this is just an overview of what we're going to be talking about in the next couple of seminars. So we're going to talk about the basic principles of resolution, what limits it, and why we need this thing called super resolution microscopy. We're then going to have a bit of a kind of deep dive into the three main techniques that are available to us, and those are SIM, STED and SMLM, and I promise the acronyms will become clear as we go on. And finally, we're going to finish up by talking about where the field is at the moment, what kind of current challenges are with super resolution microscopy, and what you can expect to see in the next few years. What's the cutting edge of super resolution microscopy? So to begin with, let's just think about size scales in cell biology, and I'm assuming most of the people here are kind of cell biologists or of some description. And I really like this animation here because it shows us the full range of what we're interested in when it comes to cells. So right at the smallest end we have viruses, come up an order of magnitude, and then we have, for example, bacteria. Go through the bacteria and then we'll come up another order of magnitude, we'll have eukaryotic cells, and we'll start getting bigger and more complex. And so if we're doing fluorescence microscopy, there's a really large range of size scales that are actually interesting to us. So you might want to look at a whole cell, but we might want to look at, a very small object such as a virus or small structures within larger cells, for example, mitochondria or other organelles. And so to begin with, let's think about what actually limits the smallest sized objects that we can resolve with fluorescence microscopy. What's our limit? The thing that limits our resolution is diffraction, so something called the diffraction limit. So what I'm showing you here is a laser beam that's being shone through a slit, which you can see on the bottom. That's the slit, and this is what the laser beam looks like afterwards. Now, let's say we want to try and make this spot here smaller. The obvious thing that you would do is make this slit smaller, so close that down. And we'd hope that makes our laser spot smaller on the other side. So let's just watch this and see what happens to this spot in the middle as we close down the laser slit. So it gets smaller, but then something alarming happens and our spot actually gets much wider and spreads out again, which is somewhat counterintuitive and unexpected. And that's because what's happening is diffraction. So on the left here, if we have a plane wave, so light is a wave, we have that plane wave that's incident upon a slit that's quite big, then you just get that plane wave propagating through and just kind of truncated by your slit. However, if we start making that slit smaller, kind of towards the scale of the wavelength, then what actually happens is our wavefronts spread out, so it becomes a circular wavefront. We haven't got a plane wave incident anymore. And this happens with all sorts of waves. Any wave will do this. And here's just a really nice example of it happening at sea. So this video shows that, somewhere over here, we have the sea waves coming in, again, a straight wavefront. They're coming through this small kind of wall, and as you can see, they're spreading out in a circular wavefront. And that's exactly what's happening with light passing through a slit, but it's just a bit easier to visualise when it's a lovely beach where we can all imagine that we are, rather than learning about microscopy. So if we think about our microscope, then we have a situation like what's on the right here. So our light source in this case is a fluorophore, so there's a tiny little GFP there emitting plane wavefront, and the slit itself is actually our objective in this case. So this small aperture is actually the aperture of your objective, and that's what's causing this diffraction and spreading of the light through the microscope. So let's put a number on this. What's the smallest thing that we can see with fluorescence microscopy? So I've got our little kind of GFP here, which we imagine to be pretty much a point-like source of light. The GFP molecule itself is, I think, around 5 nanometers in size, which is very, very small, as we all know. And as it passes through the objective, what you get is not a point source of light that's 5 nanometers small, is you get something called an airy disk pattern, or which we'll refer to as the point spread function. And that's the interference pattern of diffraction through a circular aperture like our objective. And you can see it's got this central maximum and then kind of smaller maxima going out towards the edges. And the size of this central maximum is about 300 nanometers. Okay. But what limits this? Why is this 300 nanometers? Well, to explain that, we have our friend over here, Ernst Abbe, who was a German physicist with a really excellent beard. And Abbe proposed this. He said that the smallest resolvable distance between two objects in your microscope, so if you've got two things next to each other, how small can you see apart, is equal to the wavelength of the light divided by 2 n sine theta. And n sine theta, what on earth is that? Well, it's actually on your objective. It's the numerical aperture of your objective. So if we substitute some numbers in, if we've got light of 600 nanometers, so red light, and numerical aperture of 1.4, then our resolution is about 214 nanometers. Okay, so that gives us a kind of numerical representation of the smallest objects, or the smallest resolvable distances we can see within our light microscope. And just to show an example of that here, here's the structure that I've drawn. And you should be able to see that it's a pair of lines that are getting closer together. At one end, they're 800 nanometers separated. At the other, they're 10 nanometers separated. If this object was imaged with a fluorescence microscope that was limited by diffraction, as we've seen here, then you'd get something that looks like this. So above the diffraction limit, you can still resolve the two different lines. But as we get to our diffraction limit, about 210 nanometers for this simulation here, that's when these two objects are no longer resolvable as separate things. They blur into one, okay? And that's what the resolution limit is. It's where smallest distance, where you can still make out that there are two objects rather than one, for example. And you can imagine this can be really, really problematic, right? So for this simulation, you know, we have context. We're like, okay, cool. Well, I'm pretty sure this is some kind of V-shaped structure. But if we didn't have the added context of our kind of low resolution structure that we could resolve, if we just had this, you have no idea what's within that blur, which is why resolution is a bit of a problem. So here's a kind of just another schematic of our different size scales in cell biology. And what we want is we want appropriate imaging techniques for these different objects. So fluorescence microscopy is great. I love it. And it's really good because we can look at live structures, live samples. We can look at dynamics. We can get our samples to express fluorescent proteins. We have a really large palette of labelling tools available to us, loads of advantages for fluorescence microscopy. But of course, the big disadvantage is its resolution, which, as you can see, I've limited to about 300 nanometers. Of course, we also have electron microscopy, and electron microscopy is also limited by diffraction, but the way it kind of gets around this is that the wavelength is much, much smaller. So visible light uses wavelengths of hundreds of nanometers in the kind of 400 to 700 nanometer range. Electron microscopy uses beams of electrons, which have way, way, way smaller wavelengths, way smaller. And this is, of course, why electron microscopy, even though it's still limited by diffraction, can go to much, much higher resolutions, so with particle averaging down to a few angstroms in some cases. Now, the trade-off for this kind of incredible resolution in electron microscopy is, of course, that you can't use all your labels. You can't have live samples. You've got a dead sample that's gone through quite an extreme sample preparation process, and you haven't got all your lovely fluorescent proteins and dyes and labelling techniques that you have in fluorescence microscopy. So super resolution microscopy essentially exists to fill in this gap here. Sorry, before I get onto that, we can just have a quick comparison of fluorescence and electron microscopy. So, as I said, fluorescence microscopy, we've got lots of labels, it's live cell compatible, and sample preparation is pretty straightforward. We can get really nice high temporal resolution as well. We can see dynamic processes. But electron microscopy wins out massively when it comes to spatial resolution. We've got, 1, 2, 3 orders of magnitude perhaps higher resolution with electron microscopy. Okay, so what I was going to say was that super resolution essentially exists to fill in this gap here. And here we go. So super resolution techniques basically try to extend the resolving capabilities of fluorescence microscopy down to what you can achieve with electron microscopy, or kind of part of the way there. And importantly, super resolution microscopy techniques want to preserve all the benefits associated with fluorescence microscopy. So it's trying to make fluorescence microscopy approach the resolutions of electron microscopy without getting rid of all these benefits. So if we now have a look at this table and we replace this with super resolution, do we still have lots of labels we can use? Yeah. Is it live cell compatible? Yes, but in theory, and this is something that we'll come on to quite a bit throughout the next couple of talks. Sample preparation is still pretty straightforward, and the temporal resolution can suffer a bit, and of course we'll talk about why this is, but you can still get quite good temporal resolution in super resolution microscopy, down to kind of seconds maybe, which is still pretty good. But of course the thing we're all kind of looking for here is the spatial resolution, and super resolution techniques can now get us resolutions of 20 to 150 nanometers depending on what technique we're using. So that's pretty good. We're not down to the kind of real nanometer scale of electron microscopy, but we're definitely better than conventional fluorescence microscopy. So hopefully from that little intro section, you should be comfortable with why the resolution of fluorescence microscopy is limited, what limits it, and various comparisons between fluorescence microscopy and electron microscopy. Okay, great. So let's kind of get into super resolution now. So there are 3 major super resolution techniques that exist. There's a structured illumination microscopy, or SIM, stimulated emission depletion microscopy, or STED, and single molecule localization microscopy, SMLM. And there are loads more acronyms that we will come across within the next however long this takes. But these are the kind of the real big 3 that you need to remember. And they were kind of the first inklings of these kind of started in the early 90s, and they really kind of took off since then. And in 2014, I believe, the Nobel Prize in Chemistry was awarded to some of the pioneers of particularly STED and single molecule localization microscopy, Stefan Hell, William Merner and Eric Betzig. So, this is kind of all to say that these super resolution microscopy techniques are kind of a big deal. So we're going to go through them one by one and talk about how they work, what's interesting about them, when we can use them, what kind of conditions are, the operating parameters are for these. We're going to start with structured illumination microscopy. And I'm going to kind of not warn you, but admit, or kind of, own up to the fact that this is the one that I personally struggled a lot to understand. It's the least intuitive to understand, in my opinion. So kind of don't worry if it doesn't make sense. I've used this technique very often, and I still sometimes sit around and think, do I really understand this? It's a bit of a brain tease, this one. But we'll do our best, and if it doesn't make sense, don't worry, I've provided some reviews and some other resources where you can go if you do want to kind of go over it a bit slower. Okay? So before we get on to structured illumination microscopy at all, I just want to make sure that we're familiar of the concept of frequencies in images. So here's an image of, I think, mitre tracker in a fixed cell. And this is the kind of image that we're very familiar with as microscopists. This is our structure in the spatial domain. So each pixel is a physical distance, and we could associate a size to every single structure in that image. An alternative way of representing exactly the same information is in the frequency domain. So this is in the inverse or reciprocal domain. And we do that by something called a Fourier transform. And if we Fourier transform our image, and if you use ImageJ or Fiji, for example, you can actually do this to your images. It's called FFT, fast Fourier transform, if you're interested. If you do that to our structure in the spatial domain, then you'll get this really weird looking plot. And this is representing the same information, but in the frequency domain, okay? And in this plot, and of course you can get back to your original structure by performing an inverse Fourier transform on something that looks like this. So with these Fourier transforms, you can skip back and forward between the spatial domain and the frequency domain. And what this plot shows us is essentially it's plotting the different spatial frequencies in our image. So higher frequencies are towards the edge and lower frequencies are towards the middle. So a lower frequency is the inverse of distance or size. So the centre of this plot contains our large structures, and smaller structures, the finer detail, is out towards the edge. And then beyond this kind of central ring, that's where we have high frequency noise beyond our diffraction limit. And like I said, we've got this central circle where all our kind of spatial information is really contained, and that is the frequency cut of the microscope or the diffraction limit. Okay, so outside of this, high frequency noise, right in the middle, low frequency or kind of big fat structures, and towards the edge, that's our really fine detail. And to show you this, what I'm going to do is I'm actually going to show you a movie where we make images just from different parts of our Fourier domain, just to get you comfortable with the idea that it's the same information and different parts of our Fourier transform represent different size structures in real life. Okay, so here I have our Fourier image, and what I've done to begin with is I've done a inverse Fourier transform, the other F is for fast, inverse fast Fourier transform of only the frequencies within this circle. So at highest frequency, 6.51 over micron. And if I do that, you can see, yeah, indeed, this is the big fat structures. This is the low frequency information. And if I play this movie, what I do is I add more and more frequencies each time. And as I add those frequencies, what you should have seen is you get more and more detail in your image. Okay, so the further out we go in Fourier space, the more detail we get in our image. And you know, that isn't part of SIM necessarily, it's just a fundamental property of any fluorescence imaging. Okay, just to get you comfortable with the idea that images and frequencies are the same thing, they're just kind of the inverse of each other. Okay, great. What does this have to do with SIM? Oh, there we go. Just playing it again. Why not? Now that we know what to look for. Okay, so this is from one of my favourite comics, which is XKCD. And don't worry, I won't sing because this is being recorded and I don't think I need that kind of incriminating evidence in my life. But our chap over here is saying, I took a picture of my computer screen. Why is the photo covered in these weird rainbow patterns? And the very smart woman in her armchair is saying, singing, when a grid's misaligned with another behind, that's a moire. Okay, So if you're not familiar with what these two people in the comic are talking about, then it's this. So here we've got two photos of a man in a kind of quite natty striped shirt and a corrugated kind of barn door. And what you can see is these weird rainbow patterns, which are these kind of strange, almost like an oil spill type pattern over the top of this shirt and this barn door. And you don't see them in other parts of this barn image, for example. And what these kind of weird, loopy kind of fat patterns are moire interference patterns. And these moire patterns are the result of two patterns that are of a higher frequency interfering. So what are the two patterns in each case? In this case, we have the kind of stripy pattern of the shirt. That's one high frequency pattern. And when we take the picture, we've also got another pattern, right? We've got the array of pixels inside our camera. So that's our second high frequency pattern. So the structure of the shirt stripes interferes with the structure of the pixel grid in our camera. And you get these lower frequency moire fringes, they're called, this interference pattern. And the same with this barn door. Again, we've got a high frequency pattern in this image, which is the corrugated door. We've got another high frequency pattern, which is our pixels in our array in our camera. We take our photo, and as well as getting our other spatial information, we also get interference between these two high frequency patterns, our moire fringes. And here's a little video just to kind of again just show that moire fringes in these cases are annoying, but you can cleverly exploit them to find more information about your structures. So here I've got two high frequency patterns. One has some kind of structure inside it, but you can't really see what it is. And the other is just a straight grid, okay? It's just grid lines. And you can already see where I've overlapped them. You can see the moire fringes, this lower frequency information. Okay, let's see what happens if I pass one high frequency pattern over the other. You can see that in these moire fringes, you're actually beginning to get information about one of our high frequency patterns. And in this case, because I am a crazy cat lady, it's a picture of a cat. And this is really cool. So we've got our unknown high frequency pattern. We move a known high frequency pattern, just a stripe over it, and we suddenly get all this interesting information from the interference of the two. So low frequency moire patterns contain information about both of the underlying components, okay? So that's really important. It's the product of two frequencies interfering in space. So how on earth do we use this in sim to get us resolution? What you do is you think, okay, patterns. I've got one unknown high frequency pattern that I want to interrogate, and that is the distribution of fluorescent molecules within my sample. How do I generate some moire fringes that could tell us more about that? Well, let's illuminate it with a known high frequency pattern. For example, a striped illumination field. So instead of illuminating with a flat field, we illuminate our sample with a striped field. And this results in a kind of image that looks like this, which you're like, ooh, ugly, why would I do that? But this contains moire information. This contains that interference information. And if you have a little look at the Fourier transform over here, you can see something we didn't see earlier, which are these little spots in the frequency space. And this is from the moire interference. And this contains information about both patterns on a kind of high frequency level. But importantly, remember, we're not detecting outside the resolution support of our microscope. So we've brought this moire pattern in to our resolvable field, okay? And that, even though it's low frequency information, it contains information about the higher frequencies within our sample. So this is how a STIM acquisition works. It's a wide field microscopy technique, and you pass your illumination through some kind of pattern maker, okay? So that could be a diffraction grating, it could be a spatial light modulator. You can do really kind of fun things with laser interference to generate some kind of regular pattern. And you basically move the phase and the rotation of this gridded pattern and take a series of images of your structure. And so for each rotation and phase of your illumination, you get different moire patterns, and you can actually see them rotating around inferior space. So that's cool. How do we use this to increase our resolution now? We've got all these moire patterns that are the interference of our diffraction limited image of something that's a higher frequency and our grid. Well, what we can do is we can think about this as like a series of simultaneous equations. And in reality, in the sim algorithms, this is all done in frequency space. All kind of sim calculations are done in frequency space, but you don't need to worry about that. So that's all done for you most of the time. So to understand it, we're just going to think about this in real space. So let's imagine we've got our first moire pattern. We know that that is our true structure that we don't know at a very high resolution, convolved with a known illumination pattern. Okay, so we've got one equation here. It's illumination pattern 1. If you convolve that with the structure, then you get your moire pattern 1. So you know two things in this equation. Then let's say you rotate your illumination, you get a new moire interference pattern, and you again know your illumination pattern, and your structure is the same. Okay, so essentially you do this for maybe 25 images if you combine all your rotations and your phases, and essentially you get a big simultaneous equation kind of matrix where you've got your observed moire interference patterns with low frequency information. You've got one high frequency pattern that you know, and then you can use this combination to back calculate your actual structure with higher resolution than you had before. Okay, and this is what it looks like if I run all those moire patterns, those illuminated grid patterns, through the reconstruction algorithm. Then we go from our diffraction limited image here to our sim reconstruction in the middle. You can see visually, if we just look at these two images to begin with, you get a lovely increase in resolution. For example, we can have a look at the fine detail over here. You can see that we're beginning to see the lumen of these mitochondria, for example. Then if we have a look at the Fourier transform, so the frequency domain image of our sim reconstruction, you can see that we're now filling up much more frequency space, right? So this dashed circle was the original resolution limit of our image. That's where the Fourier transform information of our diffraction limit image would live. We've actually now got frequencies from further out, so we're kind of reaching further into higher frequency land, and those come out in our image as higher resolution. And our sim circle, as it were here, is actually double the radius of our diffraction limited kind of circle. So sim has doubled the resolution, which is very cool. So to recap, the key features of sim are it's a wide field technique. It can work pretty well with live cells. That's because it doesn't really care what fluorescent molecule you use. You can use fluorescent proteins very readily with sim, and the grid pattern doesn't take too long to rotate, so it doesn't take a huge amount of time to create this raw data for a sim image, which means you've got quite good temporal resolution of about 1 second normally. And multicolour imaging, for example, is also quite straightforward. Again, it's not doing anything weird with fluorescence, it's not doing anything weird with types of fluorophores. All it's doing is generating interference patterns and then reconstructing those. Okay, so those are the kind of key features of sim. Just a quick look at some of the applications of sim microscopy. There are sim microscopes available from lots of commercial manufacturers. For example, Zeiss have the Elira, Nikon have NSIM, GE/Applied Precision, I think their name is, have the Delta Vision OMX. There's lots of commercial solutions out there for sim, or you can build your own if you're feeling very bold. But this is just an example of how sim has been used. So you can extend sim into 3 dimensions. I'm not going to talk about that now for the sake of time, but it's doable. And here are just a couple of examples of how 3-dimensional and multicolour sim has been used for looking at nuclear organisation. So this is something from Lothar Schermerle's group, and it's really beautiful imaging. And you can see here what they've done is they've imaged DAPI, so staining the chromatin, and the nuclear lamina. And you can see in the confocal microscopy image, it's kind of cool. You can see the lamin kind of between some of the chromatin. If we then go to SIM, you see much higher resolution information, and you can actually see the lamina forming these little tubules between kind of chromatin globules. So that's very cool. And a more recent example, this is 3D SIM imaging showing a DNA kind of break repair protein forming a shell around a break site. So this is our little marker of a break site. This is the DNA repair protein. And to kind of protect that site from damage while it gets fixed and to make sure that mutations don't get propagated, etc., 3D SIM was used to say, actually, yeah, this other proteins and this 53BP forms an actual protective shell in 3 dimensions around this tiny DNA break site, which is very cool. So, like I said, SIM can be used pretty readily for live cell imaging. On the left here, we have an example of imaging an immunological synapse in a T cell. And what we're looking at is stably formed immunological synapse where actin is labelled with GFP. And this is SIM imaging every 800 milliseconds, so that's pretty fast. And what you can see is for example, the flow and quantified speed and direction of this actin within this immunological synapse. On the right here, this is pretty cool. This is SIM hardware that's been adapted for imaging dendritic spines in vivo. So as you can imagine, that's pretty challenging. But again, you can see the difference between the deconvolved wide field and the SIM imaging of fluorescently labelled dendritic spines in a living mouse, again, which is very, very cool. Okay. So what are the limitations of SIM microscopy? Well, one big limitation is you can only double the resolution relative to the diffraction limit. And the reason for this is because that pattern that we're illuminating with is also diffraction limited. If we could image with a beyond diffraction limited pattern, we could extend the resolution even further, but we can't. Diffraction always limits us. So if you go through all the SIM maths, which is a bit painful, then you come out the other end saying, okay, you can maximum double the resolution in SIM. Now, in theory, you can go further with techniques such as non-linear SIM, where you use saturated fluorescence to generate harmonics, and you can use photoswitchable fluorescent proteins again to get more information. But it's not classic SIM, as it were. And of course, once you start saturating fluorescence with high intensities, then you start losing the benefits of live cell imaging, for example. Another big limitation, or kind of just warning with SIM, is that the reconstructions, you put your raw images through a reconstruction algorithm to get your final image, those can be very prone to artifacts. So this is an example of a good SIM reconstruction on the left here from the data we looked at earlier. And on the right, I deliberately made some of the reconstruction parameters bad. I made bad choices. And you can see this has done kind of suspicious things to the image that you might not notice if you're not a SIM user regularly. So for example, this mitochondrial structure here, you can see in the good reconstruction, it's just one kind of tubule. Whereas in our bad SIM reconstruction, that same structure suddenly looks like two thinner intertwining structures. And that's worrying. That's something you need to be really careful of. There's another example of this here. So if you have out of focus fluorescence, for example, SIM is only kind of really good for pretty flat samples. It's not great for thick tissue. And out of focus fluorescence is one thing that can really ruin your day in terms of artifacts. And you can see in this example all these kind of little lattice artifacts appearing in the image. So that's kind of as far as we're going to go with SIM. Hopefully you should be comfortable with what happens if you illuminate a sample with patterned information and the generation of moire fringes, the basics of how SIM uses that to increase the resolution, and some of the main advantages and disadvantages of SIM. And I'm aware it is a tricky thing to get your head around. So here's kind of a few resources that might help if you want to kind of go back and sit down and look through the maths and some more in-depth about how SIM works. Okay, so the next technique we're going to talk about is stimulated emission depletion microscopy, or STED. So STED is the only confocal super resolution microscopy technique of the kind of main three. SIM and SMLM are both wide field, STED is confocal. So let's have a little refresher of confocal microscopy. In confocal microscopy, we have a laser spot that scans across our labelled sample. So this is a simulation. Let's say this is our labelled sample. We'll have an excitation beam that scans across our area. It'll be much faster than in this simulation, but you get the idea. Every time our beam passes over a labelled part of the sample, there's going to be fluorescence emitted, and you can see that here. So there's no structure there. As our beam passes over a structure, we get some emitted fluorescence. What happens to this emitted fluorescence? Well, we don't get this structure, we just get the total number of photons that came from that beam position, which go into our PMT. So low number, high number, etc. And this number gets assigned to each beam position, i.e. each pixel, in your final rendered image. Great. Hopefully that wasn't too shocking and that was nice and familiar. So what affects the resolution of confocal microscopy? Now, our scanning spot, of course, has a finite diameter because of diffraction through the optics. If you think back to that kind of slit movie that we showed, that's what's happening in our microscope. We can't make our scanning spot smaller by just making our slit smaller or objective smaller. So what does this do to our resolution? Well, for example, when our scanning beam is parked on this pixel here, even though there aren't any fluorophores beneath that position in the image, the edges of the beam are still actually exciting molecules that are quite far away. And so when we look at our image, it doesn't have zero fluorescence, it's actually got a fluorescence value. So our finite size spot means that fluorophores quite a long way from the spot centre are excited, which leads to not great resolution. Now, one of my favourite things to do in microscopy is pretend I'm the god of physics. And if I'm the god of physics, I'm going to get rid of diffraction, and I'm going to say, what happens if we just make our spot smaller? For example, so that's our limit, that's the physical limit to the spot diameter from diffraction. Okay, back to the god of physics, i.e. me for the moment. What happens if I can just make that spot smaller? What does that look like? Well, of course, if you have a smaller spot, then for the same position, for that same pixel, the spot's in the same place, but because it isn't as fat, you're not exciting fluorescent molecules a long way away, and you get a nice higher resolution image. Okay. So what STED does is, of course, we can't just make the spot smaller. Diffraction says no. What STED does is it creates the effect of a smaller spot using some clever photophysics. So in STED microscopy, we have a Gaussian-shaped excitation beam, as in confocal microscopy. So intense in the middle, quite fat, and dimmer towards the periphery. And we're just going to think about what's happening at this position here, for example. What STED does is it puts in a second laser beam that's overlapped with your confocal spot. And that's shaped like a donut, which we call the depletion beam. The STED beam, the depletion beam, same thing, it's this donut-shaped beam. And what this does is it prevents fluorescence in the intense part of this beam. Okay, so we're going to overlap these two beams like this. And so fluorescence is prevented in the intense areas of our donut beam. So in this beam periphery, we're still getting excitation from our Gaussian-shaped excitation beam at that arrow position. But then the STED beam is saying, nope, no fluorescence allowed. So you don't actually get fluorescence from that part. Of course, the only part where fluorescence is allowed from is this dim centre of the donut, i.e. the middle here, which looks a lot like a smaller spot that scans through the sample. So you only get photons detected from the central region of the scanning beam pair as it moves through the sample. So it's kind of similar to having a smaller scanning spot. So how does STED do this? How does this work at all? What's this magic that means that you can just turn off your fluorescence or suppress your fluorescence? So we're going to have a quick look at the photophysics underlying this. To begin with, we're just going to think about what's happening in the middle of our donut, so where our excitation beam is intense but our STED beam is dim. So what this kind of abstract lines meant to represent are the energy levels of our fluorescent molecule. So down here we've got the ground state of the molecule with little sub-vibrational levels, and here we've got the first excited state of our molecule, again with its sub-vibrational levels. And molecules are lazy, so before the beam hits them, they're going to be hanging out in the ground state, in the lowest energy state. So our excitation light comes in the centre of the donut, and that energy is absorbed by a molecule and it's promoted to the excited state. After a very short amount of time, we're talking picoseconds or shorter, you get vibrational relaxation, and then after a while we get spontaneous emission or fluorescence from these lower vibrational levels of the excited state back down to the ground state, and the energy that's released during that transition from high energy to low energy comes out as a photon. So that's your fluorescence that you can detect. Then we get vibrational relaxation and we're ready for another cycle. So hopefully that isn't unfamiliar. And of course, if we think about the spectrum of our fluorescent molecule, if it's a green molecule, we'll excite around the excitation, the absorption peak, for example, and then we'll detect all of this redder fluorescence, all of this longer wavelength fluorescence into our PMT to make our image. Okay, so that's exactly the same thing as happens in normal confocal fluorescence microscopy at the spot centre. All right, what about at the beam periphery? So now we have excitation light and we have STED light. Okay, so our STED beam is now intense. To begin with, it's exactly the same. So our molecule in the ground state absorbs the excitation light and is promoted to the first excited state. The STED light doesn't interact with the energy levels at this stage, okay? But what it does is it essentially sets up a standing wave between two energy levels, normally one of the lower vibrational levels of the first excited state and one of the higher vibrational levels of the ground state. So it's not absorbed by the molecule, it's not doing anything kind of in terms of energy change at this stage, but it's there in the background. It's set up this standing wave in the background. So our excitation light is absorbed, our molecule becomes excited, we get our vibrational relaxation as standard. Now this is where the STED light kind of has its effect once we've got to this stage. The STED light is basically a catalyst for one energy transition, okay? So normally, if the STED light wasn't there, the fluorescence molecule, the molecule would, at random, fall back down to the ground state. And it does that at kind of quite a random wavelength. That's why we've got a broad spectrum. It can happen at many different wavelengths and it's, pretty stochastic process. When the STED light is present, it's really kind of catalyzing the molecule to fall down one specific energy transition. And that energy transition perfectly matches the energy of the STED photon. So this is a process called stimulated emission. Our excited molecule basically resonates with our STED light. Okay, so the STED light isn't absorbed, it stays there the whole time, actually comes out the other side. So you get your original donut photon back. And it basically forces your molecule to come back down to the ground state at the same energy as your STED photon. And of course, that means that energy still needs to be emitted. So you get a second photon out from your stimulated emission that looks identical to your original STED photon. So excitation goes in, that's absorbed. One STED photon comes in, essentially two STED photons come out because you get your original photon and the one from forcing your fluorescence molecule to go down this very specific transition. And then vibrational relaxation again. So the only kind of big difference here is fluorescence microscopy can happen at fluorescence happens at any old wavelength. Stimulated emission only happens at the wavelength of your STED beam, and so you can filter that out really well. So you're still getting light out, but you know what wavelength it's at exactly. So if we have a look at the spectra, you'll have your excitation light at your excitation peak, and then you'll have your STED light somewhere in the emission spectrum tail. So your fluorescence will be between these two wavelengths, and then you'll filter out any wavelengths that are redder than your STED light, or at your STED light and beyond, because that will be basically this donut coming back out the other side. Okay, cool. So the only fluorescence you're detecting is light in the centre that hasn't undergone this STED process of stimulated emission. What determines the intensity resolution in STED microscopy? Well, the size of the hole in the centre of the donut. So what's important to know is the optics that generate your donut in STED microscopy, they're still diffraction limited, okay? You're not doing anything to break the diffraction limit in the way you make your donut. What you actually do is you make this hole in the middle smaller by just increasing the intensity of your laser that's making up the donut. So these rings are all exactly the same size, but I've just increased the intensity. So if you imagine a Gaussian or kind of a bell-shaped curve, if you increase the intensity, you're increasing the peak, but also those wings. And those wings are encroaching into the centre of that donut, basically. So we get higher and higher intensity, which squeezes this small hole smaller and smaller. And you can see this in this little animation from a paper where 24 nanometer diameter fluorescent beads are being imaged. And if you don't have your STED on, so 0 STED intensity, then you get your diffraction limited blur. If you increase the intensity of your STED beam, you make your donut hole smaller and smaller, and you increase your resolution so you can see all these individual little beads. Really cool. So the key features of STED are it's a confocal technique, and it has all those benefits of confocal microscopy. Optical sectioning, you can get very high spatial temporal resolution if you scan a very small area, and it should work with any fluorophore as long as you've got the right wavelengths for excitation and depletion. All fluorophores are capable of undergoing STED. It's like just a fundamental photophysical process, stimulated emission. Ironically, it's actually what a laser does. The acronym LASER stands for light amplification by stimulated emission of radiation. So anything can undergo stimulated emission. It isn't just a fluorescent molecule thing. Anything will STED if you try hard enough. The spatial resolution is technically unlimited. So in theory, you can make that hole infinitely small by making your STED beam infinitely intense. And it doesn't require any computational post-processing in the same way that SIM did, for example. So basically, this is a kind of turn your STED beam on, get your image, it's already at super resolution. And there are a couple of commercial implementations. So Leica make a STED microscope, and Iberia Instruments also do various STED microscopy platforms. Lots of the STED microscopy in reality has been done on home-built systems. The Iberia one's becoming very popular now, but the home-built kind of homebrew STED has classically got the best resolution so far. So let's have a look at a couple of applications of STED microscopy. So this is an example of kind of structural discovery with STED microscopy. This is two-colour STED microscopy of these two structural proteins at the neuromuscular junction in Drosophila. In the confocal image, you can see that our magenta kind of protein, this Drosophila rim binding protein, is kind of co-localized with a larger blob of this green Bruch pilot protein. If we look with STED, what you actually see is it's forming a ring around the central protein. And this could be used for actually making really exquisite kind of three-dimensional models of this protein macromolecular structure at the neuromuscular junction. Very cool. And here's an example of some two-colour live STED microscopy So this is a very cell biology application. This is looking at the G protein ARF1 and how it's involved in forming Golgi tubules. So again, our kind of Golgi apparatus is in green, and its ARF1 protein is in magenta. And you can see nice high resolution, and here it's every 10 seconds imaging of that structure. STED is also really popular for neuroscience in particular, and that's of course because we love our, we really like these advantages of confocal microscopy. We can penetrate quite deep into tissue. And here's an example of actually in vivo STED microscopy. So this is in a living mouse, and this is looking at dendritic spine morphology. So the mouse will be expressing some kind of fluorescent protein within these kind of dendrites. And here you can see really fast resolution, really fast temporal resolution, really high spatial resolution to capture actually in a living animal changes in dendritic spine morphology. Very cool. Limitations of STED microscopy. So as you might be able to imagine, multicolour imaging can get quite difficult. Remember, normal confocal microscopy, you need just one excitation beam per fluorophore. In STED microscopy, you need an excitation beam and a depletion beam for each fluorophore. Okay, and what you don't want to happen is you don't want your depletion beam of your first fluorophore to start exciting fluorescence from another fluorophore in your example, because you're going to start getting all sorts of crosstalk. So you need to be quite careful with that. And one of the, although STED has technically unlimited resolution, that requires very high laser intensities. So this is just a little example from a paper of increasing the laser intensity in STED microscopy. And what this kind of ominous yellow thing is, it shows that the sample is actually ruptured and there's leakage of the contents of the sample out. So it's actually causing physical damage to the sample because you're trying to increase the resolution, depositing huge amounts of energy into the sample. And so that can be a bit tricky. And I did some STED microscopy during my PhD, looking from a quite photophysical angle. And here's just something I did. So one of these fluorescent crystals, one over here, was imaged using STED. So you get these nice fluorescent crystals that look nice and structured, image them instead. Hopefully my face isn't in the way. It is on this screen, but not on my other screen. So apologies if you can't see this, but there's a melted crystal underneath where my face is that literally exploded because I was doing STED microscopy on it. So that's all for the first talk. Hopefully regarding STED, you should be comfortable with why a smaller beam means better resolution in general, how STED uses a combination of beam shaping and the photophysical process of stimulated emission to increase the resolution, and the advantages and disadvantages of STED microscopy. And this is really nicely reviewed in this paper here. So thank you for joining me for the first half of this talk, and I'll see you next time to look at single molecule localization microscopy and kind of future avenues for super resolution.